the Hematology Auditory Study Guide. This will be on exam 1, and this is from the lecture, The Erythrocyte. Go ahead and get started. We're working first with the red blood cell membrane and structure. What are the two main layers of the red blood cell membrane? The outer lipid bilayer and the inner protein skeleton. The outer layer is a bilayer of lipids with embedded proteins. This separates the red blood cell interior from the plasma and contains channels for ion movement. The inner layer is a protein skeleton that is responsible for the cell shape, structure, and deformability. What are the proportions of protein, lipid, and carbohydrate in the red blood cell membrane's outer layer? So I'm looking for percentages here of protein, lipid, and carbohydrate. 52% protein, 40% lipid, and 8% carbohydrate. The outer phospholipid bilayer protein complex is composed of these components and controls membrane functions such as transport, durability, flexibility, and antigenic properties. Why do red blood cells have a negative charge, and what is its purpose? They are negatively charged due to sialic acid residues attached to glycophorins. So glycophorins cause the negative charge, which prevents the red blood cells from aggregating or clumping. So each red blood cell is negatively charged, and that means that they repel one another. How is the red blood cell's internal equilibrium maintained? This is by active processes, specifically cation pumps that require energy in the form of ATP. A little bit more of a detailed explanation. The erythrocyte membrane contains cation pumps like the sodium-potassium pump and calcium-magnesium pump. These are active processes that require energy to maintain the concentration gradients of these ions, which is vital for cell integrity. What happens to a red blood cell if the sodium potassium pump fails? Sodium ions accumulate inside the cell, causing it to swell and ultimately undergo hemolysis. What is the predominant skeletal protein in the inner membrane layer? Spectrin. Spectrin is a long fiber-like molecule that provides strong and flexible scaffolding for the red blood cell. It attaches to the lipid bilayer to the cytoskeleton via the peripheral membrane protein. And I want you to go ahead and answer that. What is it that anchors spectrin to the lipid bilayer? It's anchorin. Anchorin is the peripheral membrane protein that attaches spectrin to the lipid bilayer. Red blood cell metabolism. Why are mature erythrocytes unable to synthesize new lipids and proteins? Because they lack a nucleus. Without a nucleus, the cell cannot produce the necessary genetic material to synthesize new components. This means that extensive damage to the membrane cannot be repaired, and the cell will eventually be removed by the spleen. What is the primary source of ATP for a mature red blood cell? Glycolysis. Mature erythrocytes lack mitochondria, so their metabolism is limited. Energy, or ATP, is primarily generated through glycolysis, which allows the cell to maintain its cation pumps. keep spectrin in a phosphorylated state, and maintain hemoglobin in a reduced state. So, what I just read through it, but let's repeat it. Why is ATP necessary for the cell? What are the three things that it does for the cell? It allows the cells to maintain their cation pumps, it keeps spectrin in a phosphorylated state, and it maintains hemoglobin in a reduced state. How long can a red blood cell remain viable in stored blood? Up to 7+ weeks, as long as it is an isotonic medium with glucose and adenosine. A red blood cell can continue to metabolize at 4 degrees Celsius for over 7 weeks because its energy requirements are met by glycolysis as long as glucose and adenosine are present. Red blood cell destruction. What is the lifespan of a normal erythrocyte? Approximately 120 days. As the erythrocyte ages, it becomes senescent. What are some of the changes that occur when the red blood cell becomes senescent? I'm looking for 3 answers here. What happens as the red blood cell ages? There's degradation of glycolytic enzymes, so it's no longer able to produce that ATP through glycolysis. There's damage to membrane lipids and proteins, so that membrane's starting to degrade. And the membrane becomes more rigid itself, so the skeleton's not quite as flexible. What are 2 main ways red blood cells are destroyed in disease states? Extravascular and intravascular destruction. Extravascular destruction is the premature removal of red blood cells by macrophages outside the bloodstream, similar to normal physiology. So remember, extravascular destruction is normal. Intravascular destruction is the premature death of red blood cells within the bloodstream, which is never a normal process and can be very damaging to the organ system because it's releasing the hemoglobin just freely into the blood. Where does normal extravascular destruction of red blood cells primarily occur? In the spleen. And I'm going to tell you how. Old, rigid red blood cells, because remember, they lose flexibility, the proteins and the lipids are starting to get faulty. But anyways, old, rigid red blood cells are unable to pass through the narrow endothelial slits in the spleen and are consumed by macrophages. This process is really, really beneficial because what it does is it conserves and recycles the red blood cell components like iron and globin chains for new production, and it also does not stimulate the immune system. What are key lab indicators for elevated intravascular red blood cell destruction? And remember, intravascular RBC destruction is never normal. I'm looking for 4 lab indicators. So if we start to have intravascular RBC destruction, we're going to see decreased serum haptoglobin, hemolysis in the serum, hemoglobinuria, and the presence of schistocytes. When red blood cells are destroyed intravascularly, again, never normal, hemoglobin is released into the bloodstream. This is not good. This free hemoglobin binds to haptoglobin. causing a decrease in serum haptoglobin levels. So what it does, that free hemoglobin is going to attach to the haptoglobin, and not as much is going to be free in the system, not as much is going to be measured in the lab. If hemolysis is too great, hemoglobin can spill into the urine, and that's what hemoglobinuria is. And the presence of fragmented red blood cells or schistocytes is also a key indicator. So you're just going to see the fragments of the red blood cells floating around. You'd see that on a blood smear as the schistocytes. And that's something, if we see schistocytes, you tell that to the pathologist stat, because it's never normal and that person's in a bad way. All right, hemoglobin. What is the primary function of hemoglobin? To transport oxygen from the lungs to the tissues and facilitate carbon dioxide transport back to the lungs. Hemoglobin occupies 90% of the red blood cell's dry weight. It is the protein responsible for directly carrying oxygen and facilitates the transfer of carbon dioxide. What are the three main components of a hemoglobin molecule? Iron, which is in the 2+ state, heme, and globin. So when it's in the hemoglobin, iron's in the Fe2+ state. Hemoglobin is a tetramer composed of 4 globin chains, each with a heme group. At the center of each heme group is a single iron atom. How many globin chains does a mature hemoglobin molecule contain? 4 globin chains, typically 2 identical alpha-like chains and 2 identical beta-like chains. More detailed thought process explanation. So these chains combine to form the different forms of hemoglobin. For example, hemoglobin A, or HbA, is made up of 2 alpha chains and 2 beta chains. What are the normal hemoglobin variants found in an adult red blood cell? So this is asking the different types of hemoglobin, right? Looking for 3 answers here. Hemoglobin A, hemoglobin A2, and hemoglobin F. So hemoglobin A is approximately, it's over 95%. Hemoglobin A2 is less than 3.5%, and hemoglobin F is the least at less than 1%. Less than 1 to 2%. The majority of adult hemoglobin is hemoglobin A, which is A2, B2, beta 2. A small amount of hemoglobin A2, A2 delta 2. So hemoglobin A2 is made up of A chains and delta chains, and hemoglobin F, which is alpha chains and gamma chains. What is the difference between oxyhemoglobin and deoxyhemoglobin? Oxyhemoglobin has bound oxygen, while deoxyhemoglobin does not. Hemoglobin in the lungs, high O2 partial pressure, is in the relaxed state, or R state configuration, and has a high affinity for oxygen. In the tissues, which is low oxygen partial pressure, it's in the tense or T state configuration and releases oxygen, so it has a lower affinity for oxygen. What is the term for the process that makes it easier for subsequent oxygen molecules to bind or release hemoglobin after the first one? It's called cooperativity. When the first oxygen molecule binds to hemoglobin, it causes a conformational change that increases the affinity for the remaining oxygen molecules. This ensures that in the lungs, hemoglobin can fully saturate with oxygen. And the same goes in the tissues. It can fully release in the tissues. What effect does a decrease in pH have on the oxygen dissociation curve? So, a decrease in pH is going to be more acidic. It is going to cause the curve to shift to the right, and that means that the hemoglobin has lower affinity for oxygen and wants to release it easier. So when the pH decreases or becomes more acidic, the increased hydrogen ion concentration makes it harder for hemoglobin to bind to oxygen. This decreased affinity causes the curve to shift right, which facilitates oxygen release to the tissues. This is known as the Bohr effect. How does 2,3-DPG affect oxygen transport? It decreases hemoglobin's affinity for oxygen, promoting the T state and favoring oxygen offloading in the tissues. Hold on 1 second. Let me double check this. Can take a look at that graph really quick. Ah, there's those chains. Okay. All right. So when we increase the 2,3-DPG, we see a right shift. When the amount of DPG decreases, we see a left shift. So right shift means lower affinity for oxygen. So increased DPG lowers the affinity for oxygen and it wants to unload in the tissues. All right. All right, 2,3-DPG is a small molecule that binds to the central cavity of hemoglobin in the T state. This binding stabilizes the T state, promoting the release of oxygen. The level of 2,3-DPG increases in response to hypoxia, such as at high altitudes, to allow more oxygen to be released to the tissues. Why do smokers have about 5% of their circulating hemoglobin bound to carbon monoxide? It's basically just asking, why is carbon monoxide so dangerous? It's because the affinity of hemoglobin for carbon monoxide is about 200 times greater than that for oxygen. When hemoglobin binds to carbon monoxide, it forms carboxyhemoglobin, a pathological form of hemoglobin. A single carbon monoxide molecule can fix hemoglobin in the R state, preventing it from offloading oxygen to the tissues, which is highly dangerous. We can reverse this. This is reversible with pure oxygen treatment. What is methemoglobin? Oh my gosh, methemoglobin. I need to take a break. And how is it different from normal hemoglobin? Methemoglobin is a form of hemoglobin with iron in the ferric Fe3 plus state, which cannot bind to oxygen. So remember, when in normal hemoglobin, iron is going to be in the Fe2 plus state. Yeah, and it's just going to explain that down below. Normal hemoglobin has iron in the ferrous Fe2 plus state. So ferric is bad. Fe3 plus is not good. Methemoglobin turns the Fe2 plus, the ferrous, which is good and normal, into ferric, Fe3 plus. Ferrous, like us, two people, ferrous, Fe2 plus, ferric, I don't know how to remember that. Ferric 3 plus, ferric 3. How can we remember this? Well, so Fe2 plus, there's 2 letters in Fe, iron. So that's good and normal. Fe3 plus, there are not 3 letters. So that's not the correct state. Ferrous. Ferrous is good for us. Hemoglobin, okay? So methemoglobinemia is rare and is caused by exposure to oxidizing drugs or a genetic defect. So those oxidizing drugs take the iron and take it from the ferrous Fe2 plus state and turn it into the ferric Fe3 plus state. The treatment is to add an electron back to the defective heme group using substances like ascorbic acid or methylene blue. Why is sulfhemoglobin an irreversible condition for a red blood cell? So the other two are reversible. Methemoglobin was reversible by methylene blue, and carboxyhemoglobin carboxyhemoglobin is also reversible by 100% oxygen treatment. So why is sulfhemoglobin an irreversible condition for a red blood cell? It's because a sulfur atom binds to the porphyrin ring of the heme group and the defective heme cannot be repaired. So essentially, you just need to wait until the red blood cell dies and gets recycled out. And you can get a transfusion of pure red blood cells, new ones, to try and kind of dilute the inactive, ineffective ones whenever you have sulfhemoglobin. And that is exposure to sulfur. Sulfhemoglobin is an irreversible pathological form of hemoglobin. The red blood cell carries the defective heme until the cell is removed from circulation, which usually takes about 120 days. All right,